We're really delighted to have Dr. Rahul Singh, a longtime friend and colleagues, doing biocomputing work to give us a seminar talk on using AI algorithm to advance the drug discovery for parasitic diseases. Dr. Singh graduated from University of Minnesota with a Ph.D. and also has a degree in Moscow prior to that. And he graduated slightly earlier than I did. And I always remember him as this extremely well-spoken and a hardworking student. And sometimes when I'm sneaking out in the lab at 12 o'clock midnight, he say, "Oh, it's early for you." So I think he's a he's very driven and have been for his PhD, as far as I remember, he's been doing computer vision work. And then he has the opportunity to move to the San Francisco Bay Area and become interested in applying the computer vision and algorithms to drug discovery, largely thanks to a manager, Christian Berg, who was one of the co-founder of Genback, but went to industry. So I saw his work, and he decided that the industry was good in understanding the problem, but probably was not deep enough. So he actually went back to academia, San Francisco State University, become, I think, one of the first recipient of NSF Career Award at the Computer Science Department. He has done a lot of work in the interface of disease, parasitic diseases, and the computer science. He also holds a joint appointment with the UCSD Center for Discovery and Innovation. So this topic that he's going to talk about today is of immense interest to us, because we are doing translational work, and we're of course also interested in seeing how does the biomedical informatics can combine with algorithm and AI and make a therapeutic impact. So without further ado, let me introduce Dr. Singh. Welcome.
Well, thank you so much, Jake, and thank all of you for coming. I hope you can all hear me. And so let me actually start by very briefly saying that my lab works in various areas of omics and biomedicine, and what I'm going to talk about today is one strand of the research we have been doing for the last quite a few years. And before I sort of get into the technical details of what I'm going to talk about, I want to thank all my collaborators, my students, and the funding agencies without whose help none of this would have happened. So to get going, what I'm going to talk about today are parasitic diseases and neglected parasitic diseases and techniques of finding or drug discovery against these diseases. And what I'm going to do is to use a particular disease called schistosomiasis as an example. Though I think as we proceed with the talk, you will see that the techniques I'm talking about here are applicable to other parasitic diseases as well as biomedicine in general in many, many different contexts. So the disease schistosomiasis is caused by a trematode. In this particular case, you're looking at one of the species which causes the disease, Schistosoma mansoni, and you're looking at the adult parasite which causes the disease. So on the left, you have the adult parasite. On the right, actually, you have the male and the female parasite together. So if I may use my cursor, this slender shape is actually the female, and she resides most of her adult life inside the male's body. So the male acts almost like an exoskeleton. So this is a very interesting parasite. I'm told that its normal lifespan is about 60 years. So it has very many interesting properties, which I will allude to as I discuss the disease. So the way this disease spreads is when people get in contact with infected water. So water infected with the egg parasite. And the eggs hatch in the water, and they produce a larval stage called miracidia, which go and infect the snail as a secondary host. And in the snail, they stay about 30 days and produce another larval stage called cercaria, which are released into the water. The cercaria, when a human enters the water, recognize the human skin, they bore into the skin, and then through the venous system and the lungs and the heart go and mature in the liver, where they pair the male and the female together, and they migrate to the bowel, where they stay for the rest of their lives and produce a lot of eggs. So what is very interesting about this disease is that if you look at the adult parasite, you can see it with your eyes, so it's not microscopic. And normally when you have a foreign entity of that scale enter your body, your immune system would respond to it. But the adult parasite is able to modulate the human immune system, so we don't actually respond to it. The eggs don't have this ability, the eggs of the parasite. So the human immune system responds to it and causes various pathophysiologies. So in particular, one of the most common things you notice is fibrotic granulomatous. So this is basically hardening of the tissue of the liver due to collection of inflammatory cells in response to the eggs. You can have other issues such as, for example, portal vein occlusion, ascites, and most importantly, this parasite, when it goes and lives in the bowel, it absorbs a lot of the nutrients people are taking. And most of the people who get infected by this disease are poor people. And because of the presence of the parasite in the body, it impedes development, particularly in children and women. So this is a parasite which sort of exacerbates the cycle of poverty. Fascinating mix of how disease and economics actually come together. And this is, by the way, a very broadly spread parasite. So this is a map from a decade ago, a couple or so. And you can see the various areas the parasite inflicts, particularly sub-Saharan Africa. And sort of, the number mentioned in this map is 250 plus million people who are infected. So actually the number of people who are at risk with the parasite is nearly 1 billion. So it's a very broadly spread parasite. There is a drug which can be used to treat this parasite. It's called praziquantel. This is the only drug which is used for treatment. It's a good drug, actually, as drugs go. So the positives are a single oral dose suffices. It has few side effects. It has bioactivity against the 3 major species causing the disease, and it's a cheap drug. On the negative side, the first problem is that when you have such a broadly spread disease and you're treating it with a single drug, you have the risk of drug resistance. And actually, there have been studies in the last few years coming, especially from Africa, showing that the parasite has started to develop resistance against praziquantel there. Furthermore, the drug is of partial therapeutic use in the sense that it is effective against the adult parasite, but it is markedly less effective against the juvenile stage of the parasite. So what happens is that it kills or flushes the adult out of the body, but the juvenile parasites then become adults, and the cycle of the disease continues. So because of these reasons, both the WHO and NIH have declared this disease to be one of the critical diseases for which new therapies are needed. So I'm going to now make a shift to the process of how we screen drugs against diseases. And in particular, the question I'm interested in looking at is how do we quantify the efficacy of a compound against a disease or a target at the screening stage. So efficacy of compounds against a disease is a very broad question. It has lots of different aspects, but we are looking here at the very early part of the thing. So bottom line, what you do is you try to measure the binding of the ligand to the receptor. And typically what you do is you excite a fluorescent labeled ligand using polarized light, and depolarization of the emitted light is measured to give a sense of how well the binding is occurring or how much binding is occurring. And information, this number, is plotted on a curve, which looks like this S-shaped curve over here. This is called a dose response curve. You're measuring the response, the binding against the concentration of the compound. And then you try to characterize this curve, typically with a single number, which is at the middle of this curve over here. This number can be called EC50 for effective concentration 50%, IC50 for inhibitory concentration 50%, or LD50, lethal dose 50%, depending on the context of the screening. So The problem we are going to look at, and this is a video which gives you a sense. Now, what you see in this video is not the Schistosoma winsoni parasite, but it is a parasite called Brugia malayi, which is the etiological agent of elephantiasis. And what you're seeing here is a plate with wells containing the parasite, and each well having been treated by a different compound. And what you may notice is that the parasites are responding very differently to different compounds. Some of them, for example, are completely still. Some of them, have got it knotted up completely. Some of them are moving faster. Some of them are moving slower. So there are clear, distinct, complex, high-dimensional phenotypes. And the question for us is, how do we measure efficacy if we don't have a single number like binding, but you have this very complex response to a drug by the agent we are studying? Another example of this situation is actually our own case, the Schistosoma winsoni parasite, and this is the juvenile stage, the schistosomular stage, or the somular stage of the parasite. And this is actually a video. These parasites are moving very slowly, but you can see that they are moving if you notice these guys, for example, over here. But what is very interesting to note here is that all of these parasites, this is in a single well, have been treated with praziquantel, and the video is being taken 7 days after exposure. And what you can notice is that the parasites have very diverse or heterogeneous response to the drug. So in either of the cases, what we have is a multidimensional response. Additionally, the multidimensional response is complicated by heterogeneity of response. And what we want to do is we want to figure out how can we measure efficacy here. So this scenario I'm drawing out here, is classically called, is classic in what is called phenotypic drug discovery, or whole organism screening, or a systems approach to drug discovery, where you're trying not to target a particular molecule and knock it out, but you're trying to see the holistic effect of a compound on the disease system. So the problem formulations we are interested in are the following. So first of all, what we want to do is we want to see if we can screen a very large number of compounds against the parasite in this whole organism phenotypic screening or the systemic approach. And we want to take this complex multidimensional phenotype of the parasites and try and figure out how to compute a dose response curve and come up with an EC50, IC50, or an LD50 type of a value. Okay. The second problem we are interested in is focused on the fact that parasite populations, as you just saw, have highly or can have highly heterogeneous response to a particular chemical which they're being exposed to. And what we want to find out is how can we model and compare the heterogeneous phenotypic responses of populations to a perturbation, and in this case, a chemical perturbation caused by the drug. Right now, obviously, as you can see, these formulations have much broader implication. We could do this, in human population level studies and so on and so forth. But that's that's a later story. But we're going to focus on the parasites for now. So the obvious thing, when I was introduced to this problem was that, well, you have these parasites here exposed to some drug. So what you do is you have these images of videos of these parasites from high content screening devices. So what you do is you separate the parasites from the background. In computer vision and image processing, it's called the problem of segmentation. So you segment the parasites out, maybe you measure the parasites, and then somehow you figure out the solution to the problem. And the problem of segmentation is a long studied one in computer vision. So when I first got into this problem, I said, well, this is the easy step. And hence the title of my slide. So science always surprises you with its complexity, and nature surprises you with complexity. So computer science has this rich toolkit of different segmentation techniques, especially in the context of biomedical segmentation. And the particular techniques are less important, but what I'm showing you here are a few different state-of-the-art techniques. And you notice that none of them do a very good job in segmenting parasites. Actually, some of them do a very bad job in doing this. And this reason is because, in the kind of assays which are being run here, the transition of the background to the parasite is very weak. It is not a very clear-cut transition, as you can see here, the very diffused edges, so to speak. There are lots of touching parasites. The effect of the drug is not known a priori. So for example, here is one case, and here is another case, and here is another case, very, very different types of outputs. So traditional segmentation methods, and I may add even segmentation methods based on deep learning, the more modern approach to things, have empirically, we have found that they don't work very well. So the simplest solution, the most obvious solution I thought to the first step of the problem actually didn't exist. So the way we decided to solve this problem is based on a very interesting result, which was published by Maria Conchita Moron, who is a professor in the University of Pisa, in the journal Nature in 1986. And what Professor Moron observed is the following, and I'm going to use these two diagrams here to demonstrate the idea here. So over here, what you have is a square waveform shown in this, the blocked lines. And over here, you have a triangular waveforms shown with this blocked or solid lines. And then in these dotted lines, you have what is called the Fourier decomposition of the signal. So using the Fourier theorem. And what Conchita Moron found is that features in an image, so in the case of the square waveform, the critical feature is when the bottom part transitions into the top part. And in the case of the triangular waveforms over here, the critical feature is the top edges of the triangles. So what she found is that these critical features, when they occur, at those points, all the Fourier components are in phase. So if you look at the dashed lines, you'll notice that all of them are in phase at this point. Similarly over here, all of them are in phase. They are all making this corner. And she came up with this idea that, when humans perceive important features in an image, then the Fourier components of the image are in phase at that point. And this idea is called phase congruency. And this is a very interesting idea which provides an absolute measure of significance, which is invariant to illumination and magnification of the images for finding features such as edges, which is what we want to do to separate the parasites from the background. Okay. Now, I'm going to skip some of the math over here and summarize what these points are saying by saying that you can compute the phase congruency in many different ways, but most of them don't work for our problem domain. And during the Q&A, we can talk about this if there is interest. But there is a kind of transformation called wavelet transformation, and particularly a type of wavelet called the logarithmic Gabor wavelets, which can be used to obtain the phase congruency of an image in a very precise, very localized manner. So here, what I'm doing is I'm showing you the phase congruency of an image computed using a log Gabor transformation. And here are two parasites which are touching each other. And if you notice, it's very hard even for a human expert to figure out where is the touching boundary. And I've taken this pixel, which is right on the boundary. And what I've done here is I've computed classical techniques which are used to compute edges. So for example, here is the intensity of the image. In general, an edge would represent a point where the background intensity changes to the foreground intensity. In this particular case, because of the diffused edges and the issues I talked about earlier, this doesn't always happen very cleanly. And you can notice that the intensity doesn't show any particular characteristic at this particular point. This is the point we're talking about here. Over here in this purple dotted and dashed line is the image gradient. The image gradient also doesn't seem to be very indicative of an edge over here. Over in this dotted line is something called the Laplacian of the Gaussian. So this is a classical way in computer vision to find edges. That also doesn't show any particularly interesting property. And here in the yellow, you see the phase congruency. And the phase congruency locally does get its maximum precisely at this edge point. So you can see why phase congruency could be a very powerful way of segmenting our parasites in this very complex image domain. So here is a section of the image showing some parasites. Here is the phase congruency response of the parasites. And what I've done is I've isolated a particular parasite, this one over here, and sort of zoomed into it. And now you can see a problem. The response of the phase congruency, the log Gabor filters, is not a simple thin edge of this parasite as we would intuitively expect, but a diffused, thick sort of a halo around the parasite. And this is because the phase congruency which is being computed is not sufficiently localized to define the classical edge, something like this, which is what we would expect. So it turns out that while phase congruency is very good, very interesting, it has this problem.
So can
So how do we go about solving
question, question, Rahul?
Yes, please, of course.
So you mentioned that deep learning would not work. So I was wondering if you train the deep learning with the shape of this parasite, which is probably not going to be the dumbbell shape. So in theory, you should actually achieve similar result. And then why is it challenging?
Yeah, so I think that's a very good question. So very briefly, and we can go into a more detailed discussion maybe at the end. So very briefly, the reason deep learning doesn't work is, as you know, deep learning requires a lot of data, and the data has to be representative of what you see. Now here, as you saw, I'm going to just go back a few slides. In this composite image over here, the response of this parasite to drugs is very diverse. So unless you really have screened millions of compounds, and remember, without automation, that's that sort of a screening has never been done. You do not have enough data to properly train your deep learning models. So that's fundamentally the reason. But if we had this, yes, we could. But no one has this empirically. Wherever we have tried to do this with smaller data sets, it turns out that it has trouble with kind of edge distributions of features it hasn't seen before. So, so going back, so what we want is we have this diffuse response, and we want to get this idealized sort of an edge. And here, what comes to our aid is, a theorem from, computer science, which is called the superpositioning principle. So it turns out that there are some operations in an image where what you can do is you can run the operation at different quantizations of the image, put the results back together, and get the original result back. So let me explain this very quickly with this example of thresholding. So if we have an image with these pixel values 0, 3, 2, and 4, and we threshold this image. So basically we put a 1 if the number is greater than or equal to the threshold, and 0 otherwise. So here is a threshold value 0. So you see all of these numbers are going to be 1s because they're all greater than or equal to 0. Here's a threshold value of 1. So this is a 0, all the other ones are 1s, and so on and so forth. And then if you add, if you compute the sum from a equals 1 through 4 in this particular case, you will actually get the original image back. So in this particular case, 0, 0, 0, 0 gives us a 0, 1, 1, 1, 0 will give us a 3, 1, 1, 0, 0 will give us a 2, and so on and so forth. So this is showing you the example of the superpositioning principle. And the operation of morphological thinning in image analysis. So morphological thinning is when you have a pixel distribution and you reduce pixels till you get to the middle of this distribution, like in the case of this X over here. So this thinning operation actually follows the linear superpositioning principle. And that gives us a way to solve this problem. So what we do is we take the phase congruency response, we quantize it at different levels. We take each of the levels, we apply thinning on it, and we sum up the results. And that's what you get over here, which is very close to a thin edge of this parasite. And one interesting point when you do it like this is that the middle of the distribution of these values, what is called the ridge of an edge, is localized in terms of the common barycenters across all the principal of all the phase congruency weights. And this is a different way of thinking about an edge than classically, which is done in computer vision. So when you do this, you get this near thin edges. And we do one other operation, which is called, we binarize this image using something called hysteresis. So we locate pixels which are above a high threshold, which is automatically determined by us, and or above a low threshold, but are connected to pixels above the high threshold. So we get this information, and then we do some other processing. I'm going to skip most of it in the interest of time, which basically allows us to remove the internal structures of the parasites because we are only interested in the contours of the parasite, and we perform what is called a watershed segmentation to actually finally separate the parasites out. So let me show you an example or 4 examples of how this algorithm is working. So this is controlled praziquantel, statin, simvastatin, and chlorpromazine. And what you see here are parasites are differently colored if they were separated during segmentation. If they were not, they contain the same color. So for example, You look at these two parasites, they're clearly touching, to the human eye at least, but they have different shades of brown. So the segmentation algorithm was able to separate them. Here again, the segmentation algorithm is able to separate them. Doesn't always work. So for example, here, the segmentation algorithm was, it took these 3 touching parasites, was able to separate this one out, but could not separate these 2 out, and so on and so forth. Okay, so this is just a visual example of what this is doing. So more quantitatively, we did precision recall studies. So we took 200 images containing 8 different positions of 870 unique parasites with 1712 instances of touching parasites. So we went and manually figured all of these things out in the data. And then we computed the precision and recall. So precision being the probability that a segmented pixel is indeed from the parasite, from the foreground, and recall the probability that a foreground or a parasite pixel is indeed recognized to be such. And we got precision of 0.95 and a recall of 0.946 on this data set. More interestingly, over here in the bottom here, I have highlighted this is a measure called the object count discrepancy. So what this does is it has a human expert very carefully hand segment the parasites. And it takes the automatic technique, looks at the parasite segmented by the automatic technique, and finds out on an average how different it is from the human segmentations. So the human segmentations took people, a couple of days to do. The automatic segmentation was instantaneous, and the difference between them was about 4%. So this is the error, one way or the other of this automatic segmentation. All right, so now we have solved the segmentation problem, and we can actually get to the problem we were interested in, which is how can we take the heterogeneous response of these parasites, high-dimensional heterogeneous responses, and compute this dose-response curve and some sort of a single value which characterizes this curve. And to do this, we reformulated or repositioned the problem in the following way. So what we are really talking about here when we are talking about how the parasite has been affected is that each concentration, we're going to look at the control, we're going to look at the exposed parasites and figure out how many of the exposed parasites differ from the control at that particular concentration. Right? So if we think like this, or if we reformulate the question in these terms, we get to what is a quantal formulation. So at each point in this quantal formulation, at each concentration point, what we're interested in determining is the fraction of the parasite population that can be considered to be affected by the compound as compared to the controls. And the moment we phrase the problem like this, this becomes a classical classification problem in machine learning. So we're trying to separate the affected parasites from the control parasites. And we have many ways of solving the classification problem. So we thought we are done. And again, nature disappointed us. So we tried to run, in this particular case, a classification algorithm called support vector machines on feature descriptions of the parasites. So basically, the parasites were represented by 75 different features, which were capturing their shapes, their intensity, their texture, and so on and so forth. And we didn't do so well. And we were quite surprised why we didn't do so well. So I'm going to take this figure over here to try and explain the problem to you. So what I've done here is I've taken the 75-dimensional feature space of the parasite and mapped it to a 2-dimensional principal component representation. And this representation is not very good. It's only capturing 20% of the variance in the data. But even with 20% of the variance, it suffices for me to show you the problem. So what we have here in little circles like these ones, empty circles, the normal parasites. In empty triangles like over here, we have affected parasites. Little small filled circles are the control parasites for each of the experiments. So this study actually has combined all the experiments we had. This has 11,309 parasites in there. And with larger filled circles, I'm showing you the centroids of the controls. Okay. Now, one of the things you notice here, and by the way, this line is the separation boundary, which we computed, which I'm going to talk about later, how we computed. And incidentally, it does reasonably good job separating the affected parasites from the non-affected parasites. But the point to note here is look at the controls. Now, the controls, even with 20% of the variance captured, are spread all over. Right? So it's not that the controls are some nice one location of the feature space, they're spread across the feature space. So basically, this is why we were having trouble with a naive classification approach, because the control over time actually changes. So what I mean by that is when you have a group of parasites, you've exposed them to a drug and you're observing them, let's say over 7 days, the controls also change over 7 days. They're not static. And what we need to do is to find out how we can capture the so-called drift in the controls so that the separation boundaries in a classification problem can be drawn properly. So what we decided to do, let me just hide the controls. Give me a second, please. So what we decided to do was to see how a human expert actually does this thing manually, because that's what people have been doing since they've been studying this parasite. So I'm going to show using this cartoon what the human expert does. So this is day 1, for example, and here in filled green circles are our controls. The triangles represent affected parasites. Unfilled circles represent unaffected parasites, and the red filled circles in this cartoon are representing degenerate parasites in the control. So these are parasites, the control parasites, which for some reason, for example, died. And this happens. Okay? So what the manual expert does is he or she would discount the degenerated controls. Then they will have the control distribution, the affected and unaffected distributions, and then they will figure out which is affected, which is not affected, i.e. they would draw the classification boundary. Similarly, if you go to the, say, for example, a few days downstream, the controls have drifted, they have changed. And the same thing happens, the expert will go and discount the degenerate controls and then draw the classification boundary. So if we want to automate this approach, so what we need to do is we need to distinguish, therefore, the normal control parasites from the degenerate control parasites. We need to come up with a new way of encoding features so that the drift in the controls is actually captured. And then we can classify the control parasites and the affected parasites to draw this decision boundary and use this learning machine for data which it has not seen, so to make predictions. So, so this allows us to come up with this 2-stage learning architecture. So in the first stage of this architecture, what we do is we could classify the control parasites as normal controls or degenerate controls. We exclude the degenerate controls, we take the normal controls and basically cluster them and find the centroids of these control clusters. Then to capture the drift in the controls over time, what we do is we take each of the feature vectors of the parasites which have been exposed to the drug, and we augment with this feature vector with the feature vector of the corresponding control. So if the control drifts, the feature vector of the control, which is now appended to the feature vector of the parasite, which has been exposed to the drug, will also appropriately drift with the control. Okay. So essentially shifts in the control are now explicitly encoded in the data. And then what we do is we construct what is called a bank of support vector machine classifiers. So based on these new features we have computed for the parasites which have been exposed to the drugs, we take 10 support vector machines which are trained using 10-fold cross-validation. And then we use them and we basically have them vote on the state of a parasite as to whether it's affected or not affected. And then we take the majority of this opinion and we classify the parasite to be either affected or not affected. And that's basically what I'm talking about here in the first point. So this is prediction by majority voting. And once we have found the number of parasites which are deemed to be affected after majority voting, we look at the quantile formulation of the problem. We compute the fraction of the parasites which are affected at any point, and this gives us, a point on this curve. And then we fit using something called the Hill equation, a curve to these points to get our dose response characterizations. So, how does this work? So I'm going to show you some experiments where I'm going to show you results of dose response studies over multiple days, over multiple concentrations for 12 compounds. In this case, the images were collected every 24 hours for 4 days. So we had 258 images containing about 11,000 parasites, and we're going to call this set, the main data set. So this data set was then split into training, which is roughly 5,500 parasites, and testing, which is roughly 5,800-ish parasites. So slightly less in training, slightly more in testing. 4 human experts were requested to manually and separately, individually classify each of the parasites as normal or degenerate. And these results would then be used by us to see how well the automatic results correspond to what the experts are doing. And of course, we do a consensus annotation using majority voting across the 4 experts. And if there are ties, then we classify the parasite to be degenerate. So here are some numbers which tell us, how well the method does in terms of sensitivity, specificity, positive predictive value, negative predictive value, and accuracy. But I think a better way to experience the results of this method is actually to see the dose response curves it produces. So here are the dose response curves. So in green dotted lines, I have the manually produced curves. So these people, these 4, PhDs, postdocs, assistant and associate professors at that time in UC San Francisco, sat over many, many days and hours to actually manually do this. And in red, we have the automatic curves, which were obtained basically instantaneously using the algorithm. So here is the one for atorvastatin, and all these 12 different drugs. And you can see in most of the cases, actually the curves very closely agree with the consensus result from the human experts. The one difference, if you note, actually is for praziquantel. Look at over here. So where in the lower concentrations there is agreement, but in the higher concentrations, the predicted results and the manual results start to differ. And this is this curious case. So what happens in the case of praziquantel? So what I'm showing you here actually is the fit of what the algorithm is producing with the individual predictions of the experts. And over here, I'm showing you how well the individual predictions of the experts agree with themselves overall for the data set, and particularly for praziquantel. And one of the things you notice is that for praziquantel, the experts have much lower agreement amongst themselves than they have for the overall data set. So basically, the expert calls on what is affected and what is not affected significantly differ between the experts for praziquantel as compared to the other drugs. And this is well known in this domain. So praziquantel produces phenotypes which are always very controversial. People are not sure. So there's a cautionary note. This is a machine learning approach to the problem I showed you. And because it is a supervised learning approach, if you have disagreements in the training data, these disagreements are also going to be reflected in how the algorithm works. Right? And this is the question in some sense of bias in machine learning. So here is one example. But other than this particular case of praziquantel, I'm going to go back one slide here for a second. Again, to give you this visual idea, you can see how well the automatic approach actually correlates with expert assessments of those response characterizations. All right, so let's now very quickly, in the time available, move to the second problem, which is how can we compute or compare population-wise responses to different compounds? So again, the point here is that you expose the parasite populations to a compound, and even at the fixing concentration and fixing duration of exposure, you see the parasites vary in the responses. And I forgot to mention this when I was starting. The parasites cannot be cloned, so these are not cloned. So this is very different than what you will get if you work with C. elegans or zebrafish, for example. So these are wild type, all of them, and that's what is causing the heterogeneity in the responses. So in this case, to solve this problem, again, what we do is we map the parasites to a feature space. In this particular case, we're going to use actually an 11-dimensional feature space consisting of area, perimeter, ratio of the major to minor axis, ratio of the parasite area to the bounding box of the parasite, average intensity, variance of intensity, and some measures of texture. And what we did was we took a bunch of parasites. This experiment is actually from a study we did to establish polo-like kinases, their analogs in the parasite as a established or a desirable target for drug discovery in the parasite. And we used a bunch of compounds from GlaxoSmithKline as well as internal compounds. And what I'm showing you here in this image very quickly is a principal component mapping of this 11-dimensional space. And to simplify the picture, I'm just showing you the compounds color-coded in terms of no effects, mild effects, or severe effects. And there are a lot of interesting things actually we can talk about in this picture, which we don't have time for today. But very briefly, I can point out to you that this distribution actually has a shape. So which means that the phenotypic responses of the parasites are not random. There is a structure to the phenotypic responses. Also, as the R groups in the compounds are varied, the effect of the compounds on the parasites actually has a direction. And this is very important because this shows that phenotypic studies can actually be used to do lead optimization. This is a very, a lot of times medicinal chemistry people think that phenotypic experiments don't provide enough guidance for lead optimization. This figure actually shows very briefly that this is possible. But what I want to do actually is to answer the question of how we can compare the parasite populations. And to do this, what I'm going to do is I'm going to build a network on these parasites. So I'm going to go back one slide here. I'm going to build a network on the parasites which have been exposed to the drugs. And the way I'm going to build this network is I'm going to connect parasites which are close to each other, which are similar to each other in terms of their feature vectors. So I'm going to start with a population of parasites which is not connected, where we have, we are going to compute all the feature vectors and compute the similarities between them. Then I'm going to connect each parasite with its nearest neighbor, with its first nearest neighbor. Then I'm going to connect each parasite with the second nearest neighbor and combine these two sets. Then I'm going to do this for the third nearest neighbor, combine the sets, the fourth nearest neighbor, and so on and so forth. And why am I doing this? Because what I'm trying to do is I'm trying to figure out what is the right neighborhood which tells me that similar parasites are connected. But I don't know what is the right neighborhood. So I have no way to estimate a priori how many nearest neighbors I need to go here. So I'm going to go all the way. And then I'm going to take the results and then I'm going to analyze it. Okay, so basically, I take this distribution where all of these parasites are now connected using different nearest neighbors, and I compute properties of these networks. So in this particular case, I'm showing you some of the global properties of these networks. So the particular properties we're going to look at are shortest path length spectrum, which is the distribution of all the shortest paths in the network, the degree distribution of the network diameter, the clustering coefficient, which tells us how well this network has interconnected clusters. So, for example, here is the network diameter over here. And I'm computing it for different values of K. For K1, for K2, which is K1 union K2, for K3, which is K1 union K2 union K3, and so on and so forth. So you notice what happens in the network diameter is it fluctuates till about K equals 5 over here, and then it sort of stabilizes. The path length spectrum, the variance in the medium becomes smaller as K increases. And again, after K equals 5, it takes a particular distribution and it stabilizes. The degree distribution, which I'm showing you over here, it is linear as K increases, which means that the degree is increasing linearly, the variance is increasing linearly. And the clustering coefficient, which is over here, remains constant with K. And this tells us that this is an evenly connected network and it lacks any central nodes or cliques. So having said this, so basically what this tells us is that there is a value of K equals 5 with the network properties stabilized. So we take that value and then we decompose this network using a notion called graphlets. So graphlets are these little subgraphs or induced subgraphs, and we find how many graphlets. So in particular, we are looking at 29 different types of graphlets. So these are capturing 29 different types of connectivities in the network. And we find how many the frequency of each graphlet for the network. And the network is a set of parasites exposed to a particular drug. And then when we want to compare two populations of parasites, we compare them in terms of their graphlet signatures. So basically, we are looking at how often certain connectivities occur in each of these networks. And then we compare these distribution of connectivities using a particular distance, which is defined for graphlets. I know in the interest of time, I'm not going to go into details, the mathematical details of the distance, but I'm happy to explain it during Q&A. And let me give you a very quick example to show you how this works. So here is a compound from GlaxoSmithKline, which affects the parasites, at a particular concentration of 10 micromolars. Here is another compound which affects the parasites very similarly, as you can visually see. I'm taking examples which are visually obvious. And here is yet another compound which is providing a completely different type of phenotypic response. So when we want to compare these populations, we would expect that GSK 326090 is going to be very similar to GSK 483724, and that's going to be very dissimilar to GSK 4484598 or whatever, right? And what I've shown here is this GSK 326 is this third row over here. GSK 483 is this fifth row over here, and GSK 448 is this one over here. And the corresponding colors are showing you this distance between this graphlet frequencies. And if you look at number 3 and number 5, these two, so this is 3, and you notice that the next closest is 5 over here. Similarly for 5, the next closest network is 3 over here, and the farthest network from them is indeed this network 1, which is over here in blue. So I'm just, these lines are actually plotting the distance between these populations, for these particular ones against all the different drugs they were compared against. So bottom line, as you can see from this network, from this figure, is that we can actually use the topology of the phenotypic response space to compare diverse population responses to each other. Okay, so let me stop here because we're nearly running out of time and just summarize. So what have we seen in this 45 minutes or so of my talking? So I want to point out that schistosomiasis was first identified by Theodor Belhars in 1851. So this is a long time ago. And in 2009, which is when I first started working in this area, the way people would try to classify the effect of a drug on the parasites is to use controlled vocabularies or ontologies. Now, I work in ontologies too, but in this particular case, the problem with ontologies is that they do not allow us to get quantitative data. So we can't do quantitative structure activity relationships. We can't plot those dose response curves quantitatively. And in part due to the results I've shown you in the talk today. For example, I showed you how this automatic curves could be computed. So this algorithm is actually publicly available from our lab as a software. It's called Quadric. So in this software, you can upload using a single click your control images and your images of your affected parasites, press one more button, and the dose response curves are produced for you instantaneously. More recently, just a couple of years ago, we created and made publicly available the first database which relates compounds and quantitative phenotypes of the parasites and made it publicly available. So those are very significant, I think, even though I'm biased, progressions from where things were not that many years in the past. More generally, so we are now able to support fully automated and quantitative analysis of complex phenotypes. I showed you ways in which we can reason with complex high-dimensional and dynamic data. So these methods I've shown you here in the context of screening, but would have broad applicability in many other problem domains. And finally, this is a wonderful example of how biomedicine and computer science are mutually enriching. And as a final word, everything I showed you was for the juvenile state of the parasite, the ones which are not well affected by prazoquantum. So that was the focus. But here is the adult parasite. And the adult parasite, as you can see, in terms of its appearance, in terms of its behavior, is very different than the juvenile parasite. And what we are doing now is to recreate this entire machinery for the adult parasite. And whenever downstream we are successful, we would have then finally covered all development stages of medicinal value of this parasite. So with that, let me pause and actually let me stop. Thank you for your attention, and I'm happy to take any questions.
Well, thank you very much, Dr. Singh. We're opening for questions.
There is one question.
Okay.
Talk about the first part of your talk. When when training the SVM model, there were almost equal numbers of normal and degenerated samples in the training set. If one type of sample is much more abundant than the other type, would it significantly affect the accuracy of the model?
Yes, I think balancing the data obviously is important. In the case of the support vector machines, it's less of an issue because the dividing hyperplane is defined by what I call the support vectors, which is the data which is really very close to this decision boundary. But in general, yes, I know for deep learning, for example, balancing in the data would be very important.
There's another question by another Rahul, Rahul Sharma. Yeah, go ahead.
Hi, Dr. Rahul.
Hi.
It was a great talk. My question is related to the object identification similarity. So in general, in imaging, we use object detection.
Mm-hmm.
So, since we don't have a lot of data, we use synthetic data for object detection. So we create a lot of images and then train the model and then test it on the real data.
Mm-hmm.
Is this an option for you?
So, I think the crux of the issue. So, if I may ask, I think you're probably talking here about deep learning classification algorithms.
Yes,
am I right?
yes.
Yeah.
Yes.
So I think for deep learning, because those learning machines have a very large number of internal parameters, you need to have a very large data set to ensure that you're not overfitting. You're properly actually learning those values, right? So in the studies I showed you, we're not using deep learning, we're using support vector machines. And again, for the case of support vector machines, this is less of a problem. So we do not need to create synthetic data.
Yes,
but,
that's what my question was. Like, if
so
you
I think,
use synthetic
the answer
data.
to your question really is, what is the machine learning algorithm you're using? And what are the constraints you have to deal with to ensure that you're not overfitting? So if you're using, for this very problem, if you're using some deep learning architecture, you would definitely need more data, right? I mean, you're not going to be able to train those models properly with 5,000 or 10,000.
And we can generate synthetic data for this parasites?
We haven't done that, right? I mean, we haven't tried to use deep learning for classification, primarily because support vector machines work very well, as you see. Why get into these why get into a black box learning technique when you have a very explainable learning technique which is doing very, very well? But if you had to generate synthetic data, remember, this is the data we are talking about here is the response of parasites to drugs. So that is, in my opinion, that's a much more complicated case than what you're talking about in the simple case of object recognition, right? So this is much harder to generate. You do not know often how a drug will act on a parasite. So to create a representative sample just synthetically might be, I think this is a more complex domain, might be harder to do.
Yeah, thank you.
Sure, thank you for the question.
There's a question by Dr. Semino. Should I read it or?
Please, Jake.
Okay. So how generalizable are your methods to other organisms, including single cell and viral, or things about biology of Schistosomiasis that are being leveraged?
Yeah, so we have empirically not done this, and I would love to do this. But I think, if I look at it from a conceptual perspective, there is nothing in what I showed you which is schistosome specific, right? We are not training anything other than the support vector machine. So nothing here is particularly tied to this parasite. And this parasite is just an example we have used of a significant disease. But I would love to try this on other problem contexts with let me say some anticipation of success. How about that?
Yeah. Well, I'm intrigued by your solutions. I'm actually also thinking in other contexts, there may be a need for just finding some kind of a hallmark features, and then of course from one organism to another, the hallmark features of phenotype may be different. For example, if you look at just human, right? So you may say the drug effect may not increase the overall weight, but the waist circumference will be increased. So you will have actually somehow looking at, right, 1 meters above the feet and then measure it. So is there such a thing that you do not just look at anything that is exponentially, but only focus on certain markers of phenotype depending on your problem domain? Does it make sense? Or
no, I think it makes sense. And so obviously this would be very problem specific, but any knowledge we have, I think I'm a practitioner at the end of the day. Any knowledge we know about a domain we should use. That's number one. And number two, again, I'm going to go back to this point of support vector machines simply because in a machine learning sense, this is probably important there. So if there is a feature which is distinguishing, in a support vector machine, those features are the support vectors. So the machine should automatically pick that out also. But if we already know that a particular feature is critical, we can explicitly encode it in. I don't see any issues with that. A purist machine learning person would disagree with me, and then I would respect the disagreement. But at the end of the day, I'm trying to cure diseases. So that is more important to us.
all right, wonderful. It's 10 o'clock. I know that the people may have additional question, but you can send it to me or maybe email directly to Dr. Singh.
Absolutely.
Thank you so much again this morning.
Yes, please feel free to. And I know that you guys all have to go to another speaker. I might actually join you guys there also, but thank you for coming and attending and for listening to the talk, and thank you for all the questions.
Yeah, and I want to extend my thanks too, Rahul. Thank you very much for doing this. This is very, very informative, and I thought very accessible way to look at how machine learning can be used without having to be a data scientist. I think I got it. So thank you very much.
Thank you so much, Jim.
All right, bye.
Take care then. Bye.